So how do I think about networks? Well, certainly social networks pops right up. It's been a hot topic now for a while, and it is very good way of thinking of how things might interact with each other in a more complex way. And there is, in fact, all these mathematical systems and ways of describing networks that you could use thinking about how genes might be corresponding with each other. You could use a lot of the same language that people use. I mean, in fact, I did. I picked up the word node and the word edge. Those words are very useful in just visualizing in your head how two genes might be talking to each other, or 3 or 5 or 10.
Talk me through that a little bit. What's your vision of how that exists or functions?
I sort of think of it like a system of, this is in the film also, of airplanes going and landing in different terminals, and how there are hubs that can be quite huge, and others where just a plane goes just to one little outlying location, and when it arrives, it either reinforces, so you have a plane that's flying to some distant place, and it goes back and forth and back and forth. Alternatively, that plane can fly to a distant place, and that distant place is then says, oh, okay, now I'm going to now take this piece of information to a brand new place. There's so many ways, if you just think about how airports work. It's a kind of network, and I can totally see genes functioning that way, talking to each other, sometimes turning each other on, sometimes turning each other off, and using just a very simple tiny piece of their RNA that's made by that particular gene, and having that guide a protein, an argonaute, an airplane, to another gene that's sending out its radar. It's so simple.
So the freight
it's so
is
perfect.
The guide?
Pardon me?
What is the plane, in this analogy, the plane is argonaute?
The plane is argonaute, and it's being guided by this little RNA fragment that's produced specifically by one gene to connect with others.
And what's it connecting or transporting? What is the...
Oh, I guess it's sort of like, you pick up a letter in one place, and it only connects with, or it's a zip code, and you're following the zip code to one specific other place. So it's carrying that little piece of information that this is where you should be going.
Mm-hmm. And what happens when it arrives? Sort of like, what are the consequences of that system?
Well, what's most well understood now is that the consequences of getting one of these argonaute proteins to a certain piece of DNA is that region of DNA is then sort of quieted down, is silenced. And there may be different degrees of that. It may be that DNA is wrapped around these other big ancient proteins called histones, and those histones can move, or they can stall, and they can sometimes even start wrapping around each other and forming coils that are very hard to uncoil. So if you have a gene that you don't need ever in a particular cell type, those coils will form tightly, and that's it. That DNA is never seen again in that cell. And then there's varying degrees of that. You may have a gene that you want only turned on during certain times and turned off at other times.
Could that be the same gene in different circumstances?
Yeah, exactly. In a different cell, that gene might be critical to the functioning of that cell, and in that case, that DNA has to be accessible, and the histones have to be open and rolling, and you have to be able to get your transcription factors and your RNA polymerase in there to keep things, much looser. And you could still control that gene in a manner similar to the way HIV controls its genes. You could have a dormant state where you have RNA polymerase making lots and lots of these little fragments of RNA, and then something gets triggered downline, and you get a signal that says, okay, time now for that RNA polymerase to start really rolling, and you can activate something very quickly. So you have many probably many genes are sort of waiting for the moment to be triggered, to do whatever that cell has to do to be its normal cell.
And that's where
what I think the network, each cell type, once it's finally decided that this is, I'm going to be a liver cell, it has its own networks in place, and those networks are different for the cell that's decided I'm going to be a kidney cell, or I'm going to be a brain cell, and they each have their own interlaced networks. And the development of different cell types is also one of the great mysteries, and has something to do with evolution. Has managed to have cells that can make completely different proteins, and that's pretty evolutionarily advanced. And yet each cell carries the information necessary to make all cells. How does that happen? I think it has something to do with these networks and RNAs that can turn each other on and off.